Hey biostudents, this is Mr. Wolf, and thanks for watching this videocast. In this video, we're going to talk about the amazing world of bacteria. And as you can see from the pictures around me, bacteria come in all different shapes and sizes. So let's get into it. Now before we talk about bacteria specifically, let's look at the three different domains of life. As we had talked about in class before, there are three different domains of living things: domain bacteria, domain archaea, and domain eukarya. Now domain archaea and domain bacteria are all prokaryotic cells, which means they have no nucleus. Whereas domain eukarya are all eukaryotes. They have nuclei and they have membrane-bound organelles. We belong in domain eukarya. Now what's interesting about this cladogram, this tree of life, is that we see we are very closely related to domain archaea compared to domain bacteria. And what's fascinating is that domain bacteria are the bacteria we deal with in our everyday lives. These are the bacteria that make us sick. These are the bacteria that live in our stomachs, live in our skin. We have contact with these bacteria, whereas domain archaea, these guys kind of live in more extreme environments, which we'll talk about in a little bit. Now we're going to look at bacteria structure. When we look at bacteria structure in this diagram, I'm going to work my way from the inside out. And as I had mentioned before, bacteria are prokaryotic, which means they have no nucleus, but they do have a nucleoid region. This nucleoid region is basically the circular DNA that makes up the genetic material of the bacteria. This is the one single chromosome. It's all coiled up here, but it's one circular strand of DNA that codes for all the proteins that the bacteria makes. The bacteria also have other DNA structures called plasmids. These are extra pieces of DNA found outside the main chromosome that code for other things like antibiotic resistance. They give bacteria different things like capsules and so forth. They're just basic extra genes that can be transmitted from one bacteria cell to another. We'll talk more about these plasmids when we go into bacterial reproduction. Now, like eukaryotic cells, bacteria do have ribosomes, and ribosomes are scattered throughout the cytoplasm, and they're the basic machinery that builds bacterial proteins. Their ribosomes work just like ours. They take amino acids and instructions from the RNA, from the DNA, and they build proteins. Now, the plasma membrane or cell membrane is very unique to bacteria in that they have several structures that kind of bud out from this cell membrane. One structure is called a pili. The pili are these short rod-shaped structures that kind of sit on the outside of the bacteria cell. And what these structures do, they allow the bacteria to stick to things, especially other bacteria cells. Another interesting structure bacteria have are flagellum. And these flagellum, usually bacteria have one, or they could have a couple of these long tail-like appendages that stick out that help bacteria move, especially through water. Now, the bacteria also have what are called cell walls. All bacteria have cell walls. And their cell walls are made up of a very interesting chemical called peptidoglycan. You can see peptidoglycan is pictured up here in this diagram. Now, we'll talk a little bit later that different bacteria cells have different amounts of peptidoglycan in their cell walls. But basically, this kind of protein carbohydrate complex makes the bacterial cell wall very rigid and sturdy. And finally, some bacteria have what are called a capsule. A capsule would be the outermost layer of the bacteria cell. It's kind of a polysaccharide, a sugar coating, basically, that give the bacteria some extra protection, especially bacteria that are pathogenic, bacteria that get us sick. They usually have capsules that help them evade or disguise themselves from our immune system. Now, moving on to the different domains. We're going to first talk about domain archaea. And domain archaea is found right here on our diagram. Again, these are the bacteria that are more closely related to us than we are to domain bacteria. But domain archaea, this prefix archaea, actually gets its name from the word ancient. It's not that these are the older bacteria, but these bacteria actually live in environments that may resemble what the ancient Earth might have looked like. These environments are pretty hostile compared to today's environment that we live in. Also, another thing about Archaebacteria, they have no peptidoglycan in their cell walls, which is one reason why they're put in their own separate domain, apart from domain bacteria. Now, the first group of Archaebacteria we're going to talk about are called the Methanogens. The Methanogens get their name from the byproduct of their metabolism, which is methane. Basically, these bacteria get their energy from taking hydrogen gas and carbon dioxide and converting it into CH4, which is methane. These bacteria can be found in interesting environments, like the environments found inside intestines of cows. They're also found in termites. Basically, they're anaerobic bacteria, which they don't live in the presence of oxygen. They don't need oxygen to survive like we do. Another group of Archaebacteria are the Halophiles, and this prefix halo refers to salt. It's actually Greek for salt. These are salt-loving bacteria. This ending philic means loving. So these bacteria love very, very salty environments. In fact, they can live in concentrations up to 32% salt. And if you've ever been to the ocean and ever swallowed ocean seawater, that water is about 3.5% salt. So 32% is a lot more than 3.5%. These are very, very salty environments. And as we learned in our cell unit, most cells in a very salty environment would lose water because of osmosis, but these bacteria thrive in these types of environments. These bacteria are usually aerobic, which means they need oxygen. Some use photosynthesis as their energy source, but others are just heterotrophic. And you can find these things, or these bacteria, like in the Great Salt Lake in Utah, or even the Dead Sea, where have very, very salty environments. The final group of the Archaebacteria that we're going to talk about are called the Thermoacidophiles. If you look at this word, thermo refers to heat. Acid refers to the pH of a solution, which is very low. And again, this ending philes stands for philic, which means loving. So these bacteria love very hot, very acidic environments. You can find these bacteria growing in like the hot springs in Yellowstone National Park, like right down here. What gives this hot spring its color on the edges is these Archaebacteria, these Thermoacidophiles that live there. They're also anaerobic, which means they don't use oxygen. But what's fascinating about these bacteria is that they can withstand temperatures up to 110 degrees Celsius. In fact, they don't do well at normal temperatures that most bacteria live at. These Archaebacteria can live above boiling point, which makes them very unique types of bacteria. They also can live in pHs less than 2. The pH of 2 is pretty acidic and would kill most living things except for these Thermoacidophiles. So now we're going to move on to the next domain Eubacteria, the bacteria that we deal with in our everyday life. And these bacteria by far are probably the most diverse we find on this planet. And so we're going to talk about how we classify these bacteria. The first way we classify eubacteria is by their cell shape. Now, all eubacteria are single-celled organisms. They're unicellular, but they come in different shapes. The first shape is a rod-shaped bacteria, which we use the word bacilli or bacilli to describe them. Singular would be bacillus. For example, E. coli is a bacillus bacteria. If you look over here we have a single bacillus, this rod-shaped bacteria. Another shape of bacteria, which they come in, is sphere-shaped, which we call cocci, or singular would be a coccyx bacteria, like a Micrococcus luteus is a coccyx or a spherical-shaped bacteria. If you look over here, they're basically a spherical-shaped, a circle-shaped bacteria. And the really interesting shaped bacteria are the Spirilia, or singular would be a Spirilium. These are spiral-shaped bacteria, like Spirilium voltans is a spiral-shaped bacteria. Sometimes they're referred to as spirochetes. So these are the three basic shapes. There are more shapes that we could talk about, but these are the three basic shapes of bacteria. But another way we can describe their shape is actually how they are arranged. Sometimes bacteria kind of live in groups of two, which we use the word diplo. If we look at a Dipilobacilli or a Dipilococci bacteria, you can see there's 1, 2 cells kind of next to each other. We can use the word strepto, like if you ever had strep throat. These are bacteria that are found in chains. So like this is a streptobacilli, a long chain of rod-shaped bacteria. Or you could have streptococci. This is actually the type of bacteria that causes strep throat, a long chain of spherical bacteria cells. And then we can have Staphylo. Staphylo are bacteria that kind of are grouped in clusters like this. Like if you've ever heard of staph infections, that's caused by a staphylococcic bacteria. And so those diplo, strepto, and staphylo are just ways of describing the arrangement of the three different shapes, well really two different shapes, cocci and bacilli. We really don't use diplo, strepto, and staphylo with Spirilia. They're usually free-living or single, or they stay by themselves. Now another way we can classify bacteria cells is based on their cell wall structure. And what's really hard about cell wall structure is that we really can't see bacteria cell walls very well unless we have a really powerful scanning electron microscope. So scientists have developed what's called a gram stain. It's just a staining technique that kind of identifies what type of cell wall these bacteria have. And bacteria kind of fall into two main groups. We either have gram-positive bacteria, which they have a purple color after the gram staining technique, or we have gram-negative bacteria that have a pink color after this gram staining technique. And basically the big difference between the two types of bacteria is that gram-positive bacteria have a very thick layer of peptidoglycan. I can kind of color it in right here. This very thick layer of peptidoglycan in their cell walls, which kind of holds on to that purple stain. And then gram-negative have a very thin layer of peptidoglycan kind of sandwiched in between an extra outer membrane. And this extra outer membrane is what kind of holds or keeps the pink color when this gram stain is applied. Now, gram staining is actually a 3-staining technique where you basically stain the bacteria, wash off the stain them again. Gram-positive will retain the purple color, gram-negative will retain the pink color. And the importance of knowing the type of bacteria that, especially if you have like an infection that you're dealing with, is because these different bacteria, gram-positive, gram-negative, tend to produce different types of toxins and react differently to antibiotics. So it's important for like a doctor to know if you have a gram-negative infection or a gram-positive infection, and then they could prescribe the correct type of antibiotic. If you look at this picture down here, we can definitely see there are both types of bacteria. We have gram-positive, it looks like gram-positive cocci bacteria, and gram-negative cocci bacteria here in pink. So other characteristics about bacteria is how they get their nutrition and how they metabolize their food. So basically for types of nutrition, they come in two varieties. You have phototrophs that use light as their energy source, very similar to plants that are photoautotrophic. They photosynthesize, they use light as energy. And then you have chemotrophs, which use organic or inorganic compounds as their energy source. So that's their source of energy. It's either light or it's some kind of chemical. So we use the word photo for light, chemo for chemical. Now, when metabolizing food, they also have to have a source of carbon. Just like plants use carbon dioxide to build glucose, bacteria have to use a carbon source to build the organic molecules they need. So if the carbon source is organic in a light energy using bacteria, we call it a photoheterotroph. They get their carbon from a organic source. Like we are heterotrophs. We get our energy, our carbon too, from an outside source. We don't use carbon dioxide. Whereas if they use a inorganic carbon source like carbon dioxide, like CO2, we would call them a photoautotroph because they can use an inorganic carbon to build organic molecules, kind of similar to plants. Now the chemotrophs are kind of the same way. We can use the word chemoheterotroph if they use an organic carbon source and use an organic or inorganic carbon source for energy, and we can use a chemoautotroph would use an inorganic carbon source for both energy and their carbon source. So their metabolism and their nutrition are very closely tied to either light and organic or inorganic carbon molecules. Now, when also talking about bacterial metabolism, we also have to worry about oxygen. Bacteria have different needs or uses of oxygen. So if a bacteria must have oxygen to metabolize or break down food for energy, we call them obligate aerobes. They are obligated to use oxygen, just like we are. We have to have oxygen present for glycolysis in the Krebs cycle and electron transport chain to break down glucose into ATPs. These bacteria are very similar. They must have oxygen to get ATP energy out of the molecules that they use for energy. Then you have what are called facultative anaerobes. These bacteria are interesting. They do not need oxygen, but they can live in the presence of oxygen. Oxygen will not kill them, will not damage their enzymes. Oxygen is a big electron hog, so it could cause damage to some enzymes, but facultative anaerobes, kind of like E. coli. E. coli is a facultative anaerobe. It does not need oxygen to survive, but if it's in the presence of oxygen, it's not going to damage its cells. Whereas obligate anaerobes, they cannot. They are obligated to live without oxygen. They cannot live in the presence of oxygen, like this bacteria called C. difficile, Clostridium difficile. It's a bacteria that lives in your colon, your large intestine, but sometimes, especially if you take powerful antibiotics, it gets kind of out of hand and cause some major intestinal distress, can cause some big-time infections in your body. It's very difficult to treat, which is why it has the name, or it's been given the name, difficile, because it's very difficult to treat with antibiotics. So this is a obligate anaerobic bacteria. It does not need oxygen. Actually, in fact, it cannot live in the presence of oxygen at all. It will damage its cells, or its enzymes will no longer function if oxygen is available. Now we're going to move on to bacterial reproduction. Now the way bacteria reproduce is pretty simple. Since they don't have a nucleus, they don't go through mitosis. They use a process called binary fission. Binary comes from the number 2, and fission means to split. So basically, these bacteria simply copy their DNA and they split in 2. It's a very simple process. So it's asexual because there's no fusion of gametes, there's no sperm cells and egg cells. So bacteria just basically replicate themselves. And that replication basically produces 2 genetically identical cells. As a result, because binary fission is so simple, it's very, very quick. In fact, many bacteria cells can divide or reproduce as often as every 20 minutes. So you go from 1 bacteria cell to 2 bacteria cells in 20 minutes. The next 20 minutes, those 2 will go from 2 to 4 bacteria cells, and then 20 minutes later, 4 to 8 bacteria cells. And as you can see, this is why you may feel perfectly fine in the morning, and by afternoon you might have a severe bacterial infection, because the reproductive rate of most bacteria is very, very quick, because binary fission is pretty darn simple. Now another way bacteria reproduce, in fact it's not really reproduction, but it's a way they exchange genetic material, is through genetic recombination. So recombination comes from the word recombine. So bacteria have a way of exchanging DNA with each other. This is called conjugation. Basically, those pili that we talked about can form what's called a mating bridge, where it's a structure where it kind of connects the 2 bacteria cells together, and so their cytoplasm can now transfer materials. And what they transfer are these circle-shaped plasmids. This basically helps increase genetic variability amongst bacteria cells. It helps bacteria basically evolve. You know, this bacteria A may have a plasmid that makes it resistant to penicillin. Bacteria B doesn't contain that, but if in the same environment, even if they're different species of bacteria cells, bacteria A could form a mating bridge with bacteria B, copy that plasmid with the antibiotic resistant gene, pass it on over to bacteria B, and now bacteria B is resistant to that antibiotic, that penicillin. And so this is what's helped driven bacterial evolution, which is great for them, but oftentimes not so great for us. And the final type of reproduction that we're going to talk about is called transformation. We actually did this in the lab, where we introduced a plasmid from an external source. We basically introduced this plasmid into the bacterial's environment, and the bacteria actually takes this plasmid up. It soaks up that plasmid and incorporates that plasmid as part of its genetic material. So basically, in transformation, the bacteria takes that plasmid in, that plasmid becomes part of the bacteria cell, and whatever genes are on that plasmid, that bacteria can now produce those proteins as if it was its own bacterial DNA. So we basically taken this idea of transformation and use it to our own advantage. We can produce human proteins like insulin by introducing a human gene through a plasmid into a bacteria cell. And then when that bacteria cell has its plasmid in place, it basically takes that plasmid, it copies it, and when the bacteria divides, those new bacteria cells have that same plasmid and so forth, and you get basically a colony of bacteria with that new DNA. That bacteria is basically transformed. So that's a basic introduction to bacteria. We talked about a lot of the major structures, and we talked about how the bacteria use energy, and we talked about how they reproduce. I recommend you rewatch this video a couple times to make sure you really understand the material. See you in class.